Welcome to the deep dive. Today we're diving into the Ross-Ruff-Meckert pathway,
Oh, yeah.
a fundamental signaling cascade
Right.
that's kind of like a cellular relay race
Mm-hmm.
with kinases passing the activation baton down the line.
Yeah.
You requested a step-by-step breakdown,
Okay.
and that's exactly what we're here for.
That's great. It's a critical pathway for so many cellular processes.
Right.
And when things go wrong,
Yeah.
it can lead to serious health issues,
Yeah.
like cancer.
Exactly. We'll explore how this pathway gets triggered
Hi.
by receptor tyrosine kinases, or RTKs. We'll meet the key players, highlight the feedback loops that keep everything in check, and we'll see how it all leads to changes in nuclear transcription and impacts a cell's fate. Ready to get started?
Absolutely.
Perfect. First, can you remind us why understanding this pathway is so important?
Sure.
I know it plays a role in gene expression,
Yeah.
but how does that actually affect the cell?
Well, think about thyroid hormone receptors, or TRs, for a moment.
Okay.
They're a great example.
Thyroid hormone receptors.
Yeah.
I know they're important for a lot of things.
Right.
But how do they connect to this pathway?
So TRs, they're found in the cell nucleus,
Okay.
and they directly bind to DNA.
Uh-huh.
What's fascinating is they can act as gene silencers,
Okay.
essentially blocking gene expression
Yeah.
when thyroid hormone isn't around. But when the hormone, specifically T3, binds to the receptor,
Yeah.
it's like flipping a switch.
Wow.
The receptor changes shape and becomes a gene activator.
So the presence or absence of a single hormone can completely change how a gene is regulated.
Exactly.
That's pretty powerful.
It is. And it highlights the importance of signaling pathways like the Ruff-Meckert pathway
Right.
in controlling gene expression.
Okay.
Just like TRs rely on thyroid hormone,
Yeah.
this pathway relies on a series of signals and molecular interactions to
Yeah.
ultimately influence what genes are turned on or off.
So let's get into those signals and molecular interactions.
Mhm.
This pathway is triggered by RTKs, which are like cellular antenna, picking up signals from outside the cell, right?
Right. And those signals are often growth factors or hormones.
Growth factors or
Sorry.
hormones.
Yeah. When a signal molecule binds to an RTK, it sets off a cascade of events inside the cell.
Okay.
It's like a chain reaction.
And that chain reaction is where our cellular relay race analogy comes in.
Mm-hmm.
What's the first step in this relay?
The first step is the activation of the RTK itself.
Okay.
This happens when the signal molecule, like
Yeah.
a growth factor,
Uh-huh.
binds to the receptor on the cell surface.
Okay, so the signal is received. What happens next? Does the RTK directly activate the first kinase in the pathway?
Not quite. There are a few intermediary steps.
Okay.
The activated RTK first recruits adapter proteins.
Adaptive proteins.
These proteins act like bridges,
Okay.
connecting the RTK to other signaling molecules.
Okay.
One crucial adapter protein is called GRB2.
GRB2, what does that stand for?
It stands for growth factor receptor bound protein 2.
Okay.
It literally binds to the activated RTK.
Got it.
And GRB2 has another important binding partner,
Okay.
a protein called SOS.
SOS. Is that an acronym too?
It is, but it's a bit whimsical.
Okay.
Son of sevenless.
Son of sevenless. Okay.
Don't worry too much about the name.
Got it.
What's important is that SOS is a guanine nucleotide exchange factor, or GEF.
A GEF, what does that do?
A GEF helps activate another protein called RAS,
Okay.
which is the first runner in our kinase relay.
RAS.
Yeah, RAS is a small G protein,
Uh-huh.
and G proteins are known for binding to molecules called GDP and GTP.
So how does a GEF fit into all of this?
Well, when RAS is bound to GDP, it's inactive. But when SOS,
Yeah.
our GEF, comes along,
Well,
it helps RAS swap out the GDP for GTP.
okay.
This exchange flips the switch, activating RAS
Got it.
and allowing it to pass the signal downstream.
So SOS is like a key that unlocks RAS's potential, setting
Yeah.
the whole pathway in motion.
Exactly. And from here, RAS interacts with RAF,
Okay.
the first kinase in our cascade.
RAF.
Yep. This interaction is crucial because it brings RAF closer to the cell membrane,
Yeah.
where it can be activated.
So RAS doesn't just activate RAF directly. It brings it to the right location for activation.
Right.
That's a level of complexity I hadn't considered.
It's all about precision and spatial organization within the cell.
Wow.
And this is just the beginning.
Okay.
We'll get into the specifics of RAF activation and how it passes the signal to the next kinase in the pathway shortly.
This is already so much more in-depth than I expected. Before we get to RAF, I want to go back to RAS for a second. You mentioned it gets activated when it swaps GDP for GTP. Does it stay active ever?
That's a great question. No, it doesn't.
Okay.
RAS actually has a built-in mechanism to turn itself off.
Interesting.
It has intrinsic GTPase activity,
Okay.
meaning it can break down GTP back into GDP.
So RAS can act like a self-regulating switch.
Exactly.
It turns itself on when needed
Right.
and then turns itself off
Yep.
to prevent the signal from going on indefinitely.
Exactly. And this tight control over RAS activity is crucial because too much RAS signaling can lead to uncontrolled cell growth
Right.
and potentially cancer.
That makes sense. So we have the RTK receiving the signal, then the adapter proteins GRB2 and SOS getting RAS into its active state. Then RAS helps get RAF to the right location for activation. What happens next?
That's where RAF, our first kinase, really takes center stage.
Okay.
Remember, kinases are enzymes that add phosphate groups to other proteins,
Right.
often activating them.
Yeah.
RAF specifically belongs to a family of enzymes called serine threonine kinases, meaning they add phosphate groups to the amino acids serine and threonine in target proteins.
And in the case of RAF, its target is MEK, the next kinase in our pathway, right?
You got it. But RAF's activation isn't a simple one-step process.
Okay.
It involves a series of molecular events,
Yep.
including phosphorylation and conformational changes.
So it's more complex.
It's like RAF needs to be primed and prepped
Yeah.
before it can effectively pass the baton to MEK.
So RAS brings RAF to the membrane. And then other signaling molecules are involved in getting RAF ready to do its job.
Precisely. And once RAF is fully activated, it phosphorylates MEK, triggering the next step in our cascade.
This is fascinating. It's like a carefully choreographed dance,
Exactly.
with each protein playing its part to perfection.
And speaking of MEK, it's not just a passive recipient in this dance.
Okay.
It actually amplifies the signal as it passes it down the line.
MK amplifies the signal. Tell me more about that.
So one activated RAF molecule can activate multiple MEK molecules.
Okay.
And each of those MEK molecules can then activate multiple ERK molecules,
Wow.
the final kinase in our pathway.
So the signal gets stronger as it moves down the pathway.
Right.
That makes sense.
Especially if the initial signal from the RTK was weak. It's a brilliant way for the cell to ensure a robust response,
Yeah.
even to a faint signal from the outside.
Okay, so we've gone from the RTK to the adapter proteins to RAS activating and then helping activate RAF. RAF then activates MEK, which amplifies the signal as it prepares to activate ERK. What happens next?
Well, before we move on to ERK and its journey into the nucleus, let's pause here for a moment. I think this is a good place to break and pick up the rest of the pathway in part 2 of our deep dive.
That sounds like a plan. We've covered a lot of ground already.
Yeah.
And I know there's still much more to explore in this fascinating cellular relay race. So join us next time as we delve deeper into the RAS-RAF-MEK pathway and its impact on cell fate. Welcome back to our deep dive into the RAS-RAF-MEK pathway. We've just walked through the activation of AEK, the third kinase in this cascade. What happens next?
We've finally reached the anchor leg of our cellular relay.
RK. Right. RK is the final kinase in this pathway. What makes EK special?
Well, ERK is the one that carries the signal all the way into the nucleus.
It goes inside the nucleus. Isn't the nucleus carefully guarded?
It is. The nucleus has a membrane called the nuclear envelope. That controls what goes in and out.
Okay.
But ERK has a special pass,
Interesting.
a sequence of amino acids
Yeah.
called a nuclear localization signal, or NLS.
So it's like a molecular zip code that tells the cell, deliver this protein to the nucleus.
Exactly. The
Right.
NLS on ERK is recognized by import proteins
Okay.
that act like escorts,
Okay.
guiding it through the nuclear pores, those gateways in the nuclear envelope.
Okay, so ERK gets ushered into the nucleus. What does it do once it's inside?
Remember those master regulators of gene expression we talked about earlier? Transcription factors.
Yes.
ERK's job is to phosphorylate or activate specific transcription factors.
Okay.
And this is how the pathway ultimately influences which genes are turned on or off.
So the signal that started at the cell surface with the RTK finally reaches the nucleus
Yes.
and can directly affect gene expression.
Incredible.
Yeah.
But I know this pathway isn't just a one-way street. You mentioned feedback loops earlier.
You're absolutely right. Feedback loops are critical for keeping this pathway in check.
Okay.
It's like having a thermostat
Yeah.
that regulates the temperature,
Right.
making sure things don't get too hot or too cold.
So these feedback loops prevent the signal from spiraling out of control or getting completely shut down.
Precisely. There needs to be a balance.
Right.
And one way cells achieve this is by having downstream components of the pathway regulate upstream elements.
So it's like the runners in our relay race can communicate with each other, saying, hey,
Right.
slow down or speed up. Can you give me an example of a feedback loop in this pathway?
Absolutely. Let's consider ERK itself.
Okay.
Some of the transcription factors that ERK activates in the nucleus can actually turn on genes that produce phosphatases.
And phosphatases are like the off switch for kinases, right? They remove phosphate groups.
Exactly. So in this case, ERK is indirectly triggering its own deactivation.
Oh, wow.
It's a negative feedback loop
Okay.
that ensures the ERK signal is transient, preventing prolonged activation.
That's fascinating. ERK is essentially putting the brakes on its own activity. Are there other types of feedback loops in this pathway? There are many. Some involve other kinases or phosphatases,
Okay.
while others involve changes in gene expression or even protein degradation.
Protein degradation.
It's a complex web of regulatory mechanisms, all working together to make sure the pathway functions properly. But, there's another fascinating aspect to this regulation that we haven't touched on yet. Kinase anchoring proteins, or AKPICs.
AKPs, what are those?
They're like scaffolding proteins that help organize signaling complexes. Think of them as cellular organizers, making sure all the necessary components are in the right place at the right time.
So they're not directly involved in passing the signal, but they help make sure the signal transduction is efficient.
Exactly. And what's really interesting is that AKPs can bind to a lot of different signaling molecules. Not just the kinases in this pathway. They can bind to phosphatases, other kinases involved in different pathways, and even molecules like cyclic AMP.
Cyclic AMP. That rings a bell. Doesn't it have something to do with G protein-coupled receptors?
It does. GPCRs are another major class of cell surface receptors that use cyclic AMP as a second messenger to amplify signals within the cell and in some cells. Activation of GPCRs and the resulting increase in cyclic AMP levels can actually influence the activity of the Ross-Ref-McGurk pathway.
So we have crosstalk between different signaling pathways.
Absolutely.
That adds another layer of complexity to this already intricate network.
And this crosstalk allows cells to fine-tune their responses to various stimuli. It's like having multiple communication channels open, allowing for a more nuanced and integrated response.
This is incredible. We've gone from a linear relay race to a complex web of interactions, feedback loops, and crosstalk between different pathways. It's making me realize just how dynamic and intricate cellular signaling truly is.
It really is. And all of this complexity ultimately serves to control one of the most fundamental aspects of a cell's life, its fate.
Right, we talked about how this pathway influences gene expression. But how does that translate into actual decisions about a cell's fate? Whether to divide, differentiate, or even die?
Well, think of it this way. The Ross-Ref-McGurk pathway is like a cellular guidance counselor, helping cells navigate those big life choices by controlling which genes are turned on or off. This pathway can tell a cell whether to divide, specialize into a specific cell type, or even self-destruct.
So it's not just about relaying a signal, it's about translating that signal into meaningful actions that determine a cell's destiny. Can we break down each of these cell fate decisions and how the pathway is involved?
Absolutely. Let's start with cell division, or mitosis. It's essential for replacing old or damaged cells, growing tissues during development, and repairing wounds. And the Ross-Ref-McGurk pathway is a key regulator of this process.
I can see how that makes sense. This pathway influences gene expression, and many genes are involved in controlling the cell cycle. That intricate dance of DNA replication and cell division.
Exactly. For example, the Ross-Ref-McGurk pathway can activate transcription factors that promote the expression of genes needed for DNA synthesis and cell division.
So it's giving the cell a signal to go ahead and divide.
It's like giving the cell the green light to proceed through the cell cycle.
So the pathway is like a cellular gas pedal, pushing the cell towards division.
That's a great analogy, but just like a car needs brakes, cells need mechanisms to control their division too. There are other signaling pathways, often acting in opposition to the Ross-Ref-McGurk pathway that help keep cell division in check.
So it's all about balance, making sure cells divide only when necessary, and not in an uncontrolled way. That makes sense, especially when we consider what happens when this pathway goes wrong in diseases like cancer.
You're absolutely right. Uncontrolled cell growth is a hallmark of cancer. And the Ross-Ref-McGurk pathway is often disrupted in cancer cells. But let's not get ahead of ourselves.
Huh?
Before we delve into the connection between this pathway and cancer, I think it's time to move on to the next major cell fate decision: differentiation.
Differentiation, the process by which cells become specialized.
Exactly. Think of it as cells choosing their career paths.
Yeah.
They might become neurons, muscle cells, skin cells, or any of the countless cell types that make up our bodies.
And how does the Ras-Meckrick pathway guide these career choices?
In many cases, activation of this pathway is required for cells to differentiate into specific cell types. It's like the pathway provides the instructions, the blueprint, for cells to follow as they specialize.
Can you give me a specific example?
Sure. Consider neuron development.
Okay.
The Ras-Meckrick pathway is activated by specific growth factors that promote neuronal differentiation.
Okay.
This activation leads to changes in gene expression,
Right.
turning on genes that are required for neurons to develop their characteristic structures and functions.
So the pathway helps shape the cell into a neuron by activating the right genes at the right time. That's fascinating. And in other cell types, this pathway could be driving the formation of muscle fibers, the production of hormones, or the development of specialized structures.
Exactly. The versatility of this pathway is incredible. But remember, it's not always about promoting growth and differentiation sometimes. The pathway is involved in a cell's decision to, well, end its own life.
You're talking about apoptosis, programmed cell death.
Precisely. It might seem counterintuitive, but cell death is just as important as cell division and differentiation.
Right.
It's essential for eliminating damaged or potentially harmful cells, shipping tissues during development, and even playing a role in immune responses.
And the Ras-Meckrick pathway is involved in this too.
It is. And this is where things get really interesting. Because the pathway can play a role in both promoting and inhibiting apoptosis.
So it could go both ways.
It's like a double-edged sword.
That seems paradoxical. How can the same pathway trigger both life and death?
It all depends on the context,
Okay.
the specific cellular environment, and the intricate web of other signaling pathways that are interacting with the Ras-Meckrick pathway.
So in some situations, it's like the pathway is acting as a cellular bodyguard, protecting the cell from harm. But in other situations, it might trigger the cell's self-destruct sequence.
That's a great way to put it. And this duality really highlights the complexity and context dependence of cellular signaling.
Right.
There are no simple on-off switches. It's all about these incredibly intricate networks of interactions and fine-tuning. But, you know, as fascinating as all this is, I think it's time we shift our focus to the more practical implications of this pathway.
Okay.
Specifically its role in human health and disease.
Okay. Let's talk about the real-world impact of this pathway. I know it's implicated in a lot of diseases. Should we start with cancer?
That's a good place to start. We've already touched on how this pathway regulates cell growth and division. And when those processes go awry, it can lead to uncontrolled cell proliferation, a hallmark of cancer.
I know mutations in the Ras-Meckrick pathway are very common in cancer. Can you tell me more about those mutations and how they contribute to the disease?
Well, remember how we described the pathway as a cellular gas pedal?
Yeah.
Mutations in genes that encode components of this pathway, especially RAS and RAF, can essentially jam that gas pedal down, leading to constitutive activation.
Oh, so the cell is constantly receiving signals to grow and divide, even when it shouldn't be.
Exactly. It's like a car stuck in overdrive.
Wow.
Unable to stop.
And these mutations aren't just rare occurrences.
Right. They're found in a huge percentage of human cancers, making the Ras-Meckrick pathway one of the most important targets for cancer therapy.
And that's where targeted inhibitors come in, right?
Precisely.
Those drugs designed to specifically block the activity of these mutant kinases, essentially cutting off the signal for uncontrolled growth.
And the development of these targeted inhibitors has revolutionized cancer treatment.
Wow.
Offering new hope for patients with various types of cancers, including lung cancer, melanoma, and colorectal cancer.
That's incredible. It's amazing to think that by understanding the molecular details of this pathway, scientists have been able to develop therapies that are saving lives.
It truly is a testament to the power of scientific research. But the story doesn't end there.
Okay.
The Ras-Meckrick pathway isn't just implicated in cancer, it's also involved in developmental disorders.
Developmental disorders, those conditions that arise during embryonic or fetal development, often leading to physical or cognitive impairments.
Exactly. And mutations in genes encoding components of the Ras-Meckrick pathway have been linked to a number of these disorders, including Noonan syndrome, Costello syndrome, and cardiofasciocutaneous syndrome.
Those names sound familiar, but can you remind me about their specific characteristics?
Well, they all share a range of developmental
Oh.
abnormalities, including distinctive facial features,
Hmm.
heart defects, and varying degrees of intellectual disability.
So mutations in the Ras-Riff-Mecker pathway can disrupt the delicate process of embryonic development, leading to these profound and often life-altering conditions. That's heartbreaking.
It is, and it underscores the critical role this pathway plays in ensuring normal development. But there's another area where this pathway is involved that we need to discuss, the immune system.
The immune system, that's a bit surprising. How does the Ras-Riff-Mecker pathway fit into our body's defense system?
It plays a crucial role in regulating immune responses.
Okay.
This includes the activation and differentiation of immune cells, the production of antibodies, and the control of inflammation.
So when this pathway goes awry in immune cells, it can lead to a range of problems, from autoimmune disorders to immunodeficiency.
You got it. Autoimmune
Okay.
disorders, where the immune system mistakenly attacks the body's own tissues,
Huh.
are often associated with dysregulation of the Ras-Riff-Mecker pathway,
Yeah.
and conversely, immunodeficiency, where the immune system is weakened or impaired,
Yeah.
can also be linked to defects in this pathway.
It's remarkable how a single signaling pathway can have such far-reaching effects on human health, from cancer to developmental disorders to immune system dysfunction.
It really speaks to the central role it plays in regulating so many fundamental cellular processes.
Right.
And the more we learn about this pathway,
Yeah.
the better equipped we'll be to understand and treat these diseases. But before we wrap up, I think it's worth revisiting a concept we touched on earlier, the crosstalk between the Ras-Riff-Mecker pathway and cyclic AMP signaling.
Right. You mentioned that the activation of GPCRs and the increase in cyclic AMP levels can influence the activity of this pathway. Can you elaborate on that a bit more?
Of course, in some cells, cyclic AMP can actually modulate the Ras-Riff-Mecker pathway,
Okay.
either enhancing or inhibiting its activity.
So it's like these two signaling pathways are having a conversation,
Oh,
coordinating their actions to ensure the cell responds appropriately to different situations.
exactly. And this crosstalk is just one example of how cells integrate signals from multiple sources to fine-tune their responses.
Right.
It's a beautiful example of the complexity and interconnectedness of cellular signaling networks. But I think we've covered a lot of ground in this deep dive.
Yeah.
And I want to make sure we have time to address any final thoughts or questions you might have.
Okay. This has been an incredible journey through the Ras-Riff-Mecker pathway. We've seen how it's activated, how it's regulated, and how it's involved in a wide range of cellular processes and diseases.
Yeah.
Before we wrap up, I have one final question for you. Given how central this pathway is to so many cellular processes, can you think of other areas beyond cancer, developmental disorders, and immune dysfunction, where manipulating its activity might be therapeutically useful?
That's a great question. It makes me think about areas like regenerative medicine, where we're trying to coax cells to repair damaged tissues,
Right.
or perhaps treating neurodegenerative diseases,
Yeah.
where neuronal survival and function are compromised.
It seems like the possibilities are pretty vast.
Yeah.
As our understanding of this pathway grows, so too will our ability to manipulate it for therapeutic benefit.
Absolutely. And it's not just about targeting the pathway directly.
No,
We can also look at ways to modulate its activity indirectly, perhaps by targeting those crosstalk points we were discussing earlier.
Like the connection with cyclic AMP signaling.
Exactly. If we can understand how different pathways converge and influence each other,
Right.
we might be able to fine-tune the cellular response in a more nuanced way.
It's like having a control panel with multiple dials,
Uh-huh.
allowing us to adjust the volume of specific signals, rather than just turning them on or off.
That's a great analogy. And this approach could be particularly useful for complex diseases, where multiple pathways are dysregulated.
It's exciting to think about the potential for new therapies that target not just individual proteins,
No.
but entire signaling networks.
It is. And we're only just scratching the surface of what's possible.
This deep dive has been incredible. I feel like I've gained a whole new appreciation for the complexity and elegance of cellular signaling.
Me too.
It's like we've glimpsed the inner workings of a cellular machine, marveling at its precision and power.
And who knows what other secrets this cellular machine holds, waiting to be unlocked by curious minds like yours.
So keep exploring, keep asking questions, and keep diving deep into the fascinating world of science.
Well said. And remember, every deep dive brings us closer to understanding the intricate symphony of life and harnessing its power to improve human health.
Until next time, I keep those brains buzzing And let us know if you need another deep dive into this amazing cellular world.